Hello students, welcome to this session. I, Dr. Rajeshri Mutha, MBBS DPB from CPS, College of Physician and Surgeon, Anauti Hospital, going to take lecture on general microbiology.
Hello students, today we will see the introduction of microbiology. So what is microbiology? It is study of microorganisms like bacteria, virus, fungi, parasites. It is called as microbiology. So first is scope or application of microbiology. So first is in microbiology there is identification of microorganisms like bacteria, virus, fungus and parasites, and then report of sensitivity is done for the treatment of the patient, and some serological reaction studies is also done for fast diagnosis of the disease, that is called as indirect diagnosis of the microorganism. So examples of that is rheumatoid arthritis test for rheumatoid arthritis, ASO test for rheumatic fever, CRP test for inflammatory disease, HBsAg test for hepatitis B, venereal disease research laboratory test for syphilis, HIV human immunodeficiency virus for AIDS acquired immunodeficiency syndrome, HCG test for pregnancy, Vidal test for typhoid fever. So these are few serological reactions for the diagnosis of the disease and then treatment of the disease. Thank you.
Let's study some scientists in microbiology. So first is Anthony van Leeuwenhoek, first scientist to observe bacteria and other microorganisms using single lens microscope. Louis Pasteur, father of microbiology, introduced sterilization techniques, described pasteurization of milk, vaccine development against anthrax, fowl, cholera, and rabies. The Robert Koch discovered anthrax bacilli, tubercle bacilli, cholera bacilli, and Koch's postulates. So in Koch's postulates, first is the microorganism constantly associated with the lesion of the disease. Second, isolate the organism in pure culture. Third, same disease must result when isolated organism is inoculated into suitable laboratory animal. Fourth, possible to re-isolate the organism in pure culture. Fifth, antibody to causative organism should be demonstrable in patient serum. Edward Jenner developed first vaccine, smallpox vaccine. Joseph Lister, father of antiseptic surgery.
Hello students, today we will see bacterial classification depending on the gram staining reaction. So gram positive are purple in color and gram negative pink in color. So cocci gram positive, they are staphylococci, streptococci, pneumococci or enterococci, and gram positive bacilli, they are divided into aerobic and anaerobic. So aerobic are bacillus spore forming and anaerobic spore forming are clostridium. Then gram negative bacteria, cocci are Neisseria, and gram negative cocci are gram negative bacilli are divided into different classifications. So one can be according to the lactose fermenting property on Meconici agar, they are lactose fermenting and non-lactose fermenting. So lactose fermenting bacteria are E. coli and Klebsiella, and non-lactose fermenting bacteria are Salmonella, Shigella, Proteus, Vibrio, Pseudomonas, Acinetobacter, etc. Thank you.
Hello students, let's study general bacteriology.
Hello students, today we will see about bacterial cell. So components, what are the components? There are coverings of the bacterial cell. So outermost covering is called slime layer, and the function of this is protection of the bacteria from antibiotics and disinfectants. Second covering is capsule. So it is gelatinous substance secreted by bacteria which becomes thick to form capsule around the cell wall, and function is protection and demonstration by India ink preparation, that is negative staining. An example of capsulated bacteria are pneumococcus and fungi is cryptococcus. Cell wall, it is outer layer of bacteria, and function, it gives shape and rigidity to bacteria. Cell membrane, the inner membrane, inside the cell wall there is a thin semi permeable membrane, and function, it controls inflow and outflow of metabolites, it gives mechanical strength to the bacteria. Cytoplasm, it is a liquid and water liquid suspension in which water and organic inorganic substances are there, and plus organelles are there like mitochondria which is for the powerhouse of energy, ribosomes for protein synthesis, Golgi complex for protein synthesis, endoplasmic reticulum, so it is a network of cells in near the nucleus for the protein synthesis. Lysosomes, they are sac-like structures present for the destruction of the foreign material. Nucleus, it is long filament of DNA coil inside cytoplasm. As bacteria is prokaryotic, the well-defined nucleus is not there, and no nuclear membrane and no nucleolus. Function of nucleus is bacterial division, antibiotic resistance to carry from one cell to another cell. Plasmid, it is a extra genetic material present, and it is function as a transfer of genetic material. Thank you. Hello students, now today we will see special structures of bacterial cell. So first is flagella. So flagella in motile bacteria, they possess one or more long unbranched filament that is called as flagella, and function is it is organ of motility or locomotion. Demonstration, it can be demonstrated by dark ground microscope or electron microscope or motility testing, that is hanging dropped preparation. Classification of flagella according to the arrangement is peritrichous flagella are all around the cell, monotrichous single flagella at one pole, amphitrichous single flagella at two poles, lophotrichous group of flagella at one pole. And here in this diagram you can see the flagella arrangement. Second structure is fimbria or pili. It is organ of attachment. Next structure is spores. So they are highly resistant, demonstrate state of this bacteria, not destroyed by boiling, and they are destroyed by or killed by only autoclaving, temperature 121 degrees Celsius for 15 minutes and 15 lbs pressure. Function, to survive the bacteria in adverse condition. Demonstration by special staining, gram stain, Zn stain. Thank you. Hello students, today we will see growth requirement of bacteria. So first is food or nutrients required for the growth of bacteria is proteins, polysaccharides, lipids, nucleic acids, organic, inorganic salts and vitamins. Then moisture is required. Then pH, it should around 7.2 to 7.6. According to that, acidophilic and basophilic bacteria, they are known, classified. Hello students, today we will see bacterial physiology, that is bacterial growth curve. So when bacterium is inoculated into a suitable culture media or liquid media and incubated, its growth follows a definite course, and that curve is called as bacterial growth curve. There are 4 stages in bacterial growth curve. First is lag phase, LAG. Second is log phase, then stationary or exponential phase, and then log phase or exponential phase, stationary phase and decline phase. In this diagram you can see these 4 phases. So what is lag phase? So immediately after inoculation on culture medium, there is no appreciate increase in number, but there is increase in size of the cells. So this is called lag phase. Log phase, after the adaptive to the new environment, bacteria multiply, and then it is also called as exponential phase. There is increase in bacterial number, bacterial count. Then stationary phase, after log phase, cell division stops because of decrease in nutrients and accumulation of toxic products, and then the bacterial count is static. And then after that, bacterial count decreases because of the death of the cells, because of the toxic accumulation, and this is called as decline phase. So these are the 4 phases of the bacterial growth curve. Thank you.
Hope you find this session useful for your understanding and for your exams. Thank you.